And I realized I've spent a lot of time getting feedback from students last year about labeling the slides. So most everything is labeled, but I realize this is one I don't have labeled. So let's just go over this here slowly and make sure that these blood vessels we've talked about, you've all got identified here. So let's start with the posterior circulation. So vertebral arteries, right? A right and a left vertebral artery. And then coming off laterally, we really only see it on this side, but this is the pica right here. Posterior inferior cerebellar artery. Coming off medially here, we've got the anterior spinal artery, a branch from each vertebral, and then we just have the anterior spinal artery right here, continuing down, really the whole length of the spinal cord, as we'll discuss here in a little bit. Okay, so the vertebrals come together and form the basilar artery. And off of the basilar artery, we have the anterior inferior cerebellar artery right here, and the other anterior inferior cerebellar artery over here. And we'll see in the lab how oftentimes there are a lot of variants and congenital anomalies, and here we can see this one comes up a lot higher than this one, and that kind of thing is common. Okay, so if we follow the basilar artery up, the next blood vessel we come to is right here, the superior cerebellar artery. Okay, it's right at the top of the circle there. Okay, and notice there's a nerve here. This nerve is the third nerve. Okay, and you can see the third nerve is sandwiched between two blood vessels. Okay, so the bottom part here is the superior cerebellar artery. Okay, the blood vessel wrapping around the third nerve is the posterior cerebral artery. Okay, so the circle of Willis, this picture, things are kind of twisted. We can't see much of what's going on over here, but the basilar artery terminates into a right and a left posterior cerebral artery. And really, we can only visualize the one here on this side very well. It's hard to see it over here. All right, and so this is the posterior cerebral artery. And notice that we said the midbrain is in here. You can't see it. It's deep. Okay, but the posterior cerebral artery kind of lies right on top of the midbrain. If you just were to put your finger into that space, you would reach in here and touch the midbrain. Right? So the posterior cerebral artery is interesting. It comes out here off the top of the basilar, and it wraps around, of course, on both sides, and goes all the way back. It supplies a lot of the temporal lobe here, and it is headed back to the occipital lobe. Most of your occipital lobe is supplied by the posterior cerebral artery. Okay, so here's kind of a color picture to show you the area that is supplied of the brain by the posterior cerebral artery. So it's the occipital lobe and the undersurface of the temporal lobe. Okay, extending maybe a little bit up here into the parietal lobe as well. Okay, so that's the PCA. So to get another close-up look here, here's the basilar artery. Okay, and so here is the superior cerebellar artery, the third nerve, and the posterior cerebral artery. Here we can see on this side and then on the other side. Okay, so that's the posterior cerebral artery. Now, the posterior cerebral artery, since it lies on the midbrain, it actually supplies most of the midbrain with its vasculature. Okay, but there also are penetrating branches, just like on the MCA. We talked about those lenticulostriate branches that supply deeper structures. The posterior cerebral artery also has penetrating branches that supply deeper structures, and this is mainly the thalamus. Okay, but also the sensory fibers that are in the posterior limb of the internal capsule. The lenticulostriate, if you look back at your handout there, I said it's the motor fibers in the posterior limb of the internal capsule. The posterior cerebral artery penetrating branches supply the sensory fibers in the posterior limb of the internal capsule. Okay, and so don't worry about, we get to neuroradiology later, but here's a CT scan, and this just shows you a stroke in the distribution of the posterior cerebral artery. This is actually the midbrain right here, and so the posterior cerebral artery kind of comes around like this. It supplies the temporal lobe and then the occipital lobe back here. So this is a stroke in that territory. All right, so there is a circle of Willis, which we can see as a communicating vascular network here at the base of the brain, and these are the blood vessels that contribute to the circle of Willis, which is actually pretty important because I see patients all the time where I'll do an angiogram study, which outlines all of the blood vessels, and that patient may have lots of plugged, occluded, narrowed blood vessels, yet they've never had a stroke. Okay, and part of the reason for that is things like the circle of Willis, where we're able to share vascular supply in different areas. And let me explain by picture what I mean by that. All right, so here in the drawing, this is the circle of Willis. Okay, so what makes up the circle then is the internal carotid artery right here, the anterior cerebral artery here. So notice the anterior cerebral artery continues out like this, but this part of the anterior cerebral artery. There's a connection between the right and left anterior cerebral artery, and that's the ACOM, or the anterior communicating artery. Okay, here's the anterior cerebral artery here, the internal carotid here. And notice we have a link between the anterior and posterior circulation that is the PCOM, the posterior communicating artery. Okay, and it joins the internal carotid artery with the posterior cerebral artery. Okay, so the posterior cerebral artery is the kind of the posterior portion here of the circle of Willis. Right, so I could just give you an example here. Let's say maybe we have a patient with some narrowing of the posterior cerebral artery right here. And so you would think, well, maybe they're going to have a stroke in that distribution. But if we've got an open posterior communicating artery here, then the anterior circulation, the internal carotid artery, can actually pump some blood supply into the posterior cerebral artery. And even though the patient has vascular disease, they don't have a stroke. Okay, and so same thing here with the anterior communicating artery. Its importance. We may have a patient with an occluded proximal portion here of the anterior cerebral artery. But if the anterior communicating artery is patent, then this internal carotid artery can actually supply both anterior cerebral arteries, even though we've got a completely plugged blood vessel right here. Okay, so that's the importance of the circle of Willis. It allows for overlapping blood supply, and patients can have vascular disease frequently without having strokes. So just another picture here of the circle of Willis and labeled blood vessels. And you can see when we actually get to the lab and we try and look at this, it's always easier to show it on a cartoon than it is. But we'll go over this, try to do this here in the lab to point out all the blood vessels. One very important relationship here. We've made the significance here of the uncus. This is the uncus right here, lying next to the third nerve. Right? Remember, if you have uncal herniation, then the uncus can push on the third nerve. But there's another important relationship, and that is between the third nerve and the PCOM, which is right here. Okay, let's just start at the beginning here. The basilar artery, here's the posterior cerebral artery on this side, the posterior cerebral artery on this side. We can only kind of see this part of it. And so, again, the link between anterior and posterior circulations is this posterior communicating artery, which is right here and which is right here. Maybe you can see a little better on this side. Okay, so the reason this is important is aneurysms, which is kind of like a little area of the blood vessel where the wall is weak and it blows up like a balloon slowly. And a very common place for aneurysms to occur is along the posterior communicating artery. Okay, so these don't cause symptoms until they rupture and bleed. But when they bleed, the blood will often compress the third nerve. Okay? So we'll talk about aneurysms later, but when they rupture, they tend to cause a very severe headache. And so if you have someone that comes in with a really severe headache and they have a third nerve palsy, then you could be very confident that you're dealing with an aneurysm that bled right in this location. Okay, so two important anatomic relationships with the third nerve, the uncus and the pecon. Okay, and here's a drawing that a student finished just last week for me, so showing you the MCA territory, which is mainly this area, the anterior cerebral artery, which again is most of the midline, but extending over here a little bit to this portion of the brain, and then the posterior cerebral artery, which is the undersurface of the temporal lobe, and the occipital lobe into the parietal lobe here a little bit. Okay, let's go over venous drainage of the brain. So we will talk here in just a few minutes about how these venous sinuses are located between two layers of the dura. So we'll go over that. But for now, let's just show the venous sinuses. So all this is labeled here. So we have a superior sagittal sinus right here, SSS, and an inferior sagittal sinus, which is right here. And actually, okay, here's one thing I didn't label, but this right here is actually a reflection of the dura that goes in between the right and left hemisphere. It's in the interhemispheric fissure. And so if you just want to write there, write falx cerebri. This whole thing right here is the falx cerebri. It's the dura projecting into the interhemispheric fissure. Okay, and so this is a reflection of the dura. And so the superior sagittal sinus lies at the superior portion of the falx cerebri. The inferior sagittal sinus, the ISS, lies on the inferior reflection of the falx cerebri. Okay, so venous blood here drains through the superior and inferior sagittal sinus, and they come together here at this area, which is called the confluence of sinuses. Sorry, one other thing I forgot to label here. Confluence of sinuses, right where my circle is. Okay, and we should back up actually here and follow the inferior sagittal sinus because it dumps into the straight sinus. So it's the straight sinus actually that joins with the superior sagittal sinus here at the confluence of sinuses. And one other vein to point out here, it dumps right into the straight sinus, is the vein of Galen, or sometimes called the great vein of Galen, which is right here. Okay, the vein of Galen drains deeper structures in the brain. Okay, so from the confluence of sinuses here, we have the transverse sinus. Okay, and this will make more sense when I can point it out over here, but let's just do this one first. From the transverse sinus, we have the sigmoid sinus right here, and the sigmoid sinus drains into the internal jugular vein. Sorry, one more you have to label there, the internal jugular vein. All right, the cavernous sinus, which we can see over here, which drains the eye and the orbital contents. Okay, the cavernous sinus joins with all of this via the superior petrosal sinus, which we can see right here. And one more you have to label, this blood vessel right here, which is the inferior petrosal sinus. Okay, so this joins here with the sigmoid sinus, and it all dumps into the internal jugular vein. Okay, now let's look at this. If we just do a cut like this, kind of look at things in a different dimension. So now this is the confluence of sinuses. Okay, so remember what's dumping into the confluence of sinuses? The superior sagittal sinus and the straight sinus, which is right here. So this comes together in the confluence of sinuses, and here we can see that the transverse sinus kind of wraps around laterally here on both sides. Here it's labeled right here, the transverse sinus. Okay, so the transverse sinuses dump into the sigmoid sinus. Okay, and then that goes out through the internal jugular vein, which I can't really show you here on this picture. Here we can see the cavernous sinus. Remember, again, that drains the eye and the orbital contents. And the cavernous sinus joins with all of this through the superior petrosal sinus, okay, which kind of joins with the transverse sinus here. They both dump into the sigmoid sinus. And the inferior petrosal sinus, which is labeled over here. The inferior petrosal sinus and the sigmoid sinus join together, and it's not shown here, but the internal jugular vein is about in this area, okay, where it all drains out. Okay, so I put the unlabeled portion in there also, so when you spent some time looking over it, see if you can label all of this on your own. Okay, and so we can do venogram studies where we can outline the venous drainage. And so here we can see the superior sagittal sinus, the confluence of sinus, the straight sinus. Okay, and here we can see, again, the confluence of sinus and the transverse sinus. And notice here we actually have an occlusion of the transverse sinus on this side. It's flowing nicely here. It isn't on this side. So if we have a venous occlusion, patients can pretty quickly get into a lot of trouble. The arterial pressure, it can't drain, and so we get increased pressure in the brain. That's quite dangerous. And so because of the increased pressure, now the arterial distribution can't adequately supply tissue, so we can get ischemic infarctions. And the increased pressure can cause leakage of blood into the brain, and that's called hemorrhagic infarctions. Okay, most often clinically we see occlusion of the superior sagittal sinus. But we can also have occlusion of the cavernous sinus. This would probably be the second most common area of venous occlusion. And remember, since the cavernous sinus drains the eye and the orbital contents, now you have what's called a painful ophthalmoplegia. Ophthalmoplegia is just a big word for the eye doesn't move very well. Okay, and so you got a lot of swelling behind the eye and restricted eye movements, and the eye gets very red and congested, and proptosis is the eyeball literally getting pushed out of the eye socket from the increased pressure. And so here's a patient. I wouldn't expect you to know what you're looking at here, but there's a cavernous sinus occlusion, and so there's a lot of swelling in the eye. The patient, can't even open the eye. If we were to lift the eyelid up, we would see he has great difficulty moving the eye. So that would be a cavernous sinus problem. Here is a venogram study. So we're kind of looking on top of the superior sagittal sinus. This is normal, but it should look like this all the way through, right? So we've got an occlusion of the superior sagittal sinus right here. And so again, that arterial flow can't drain, and so we end up getting strokes in that around that area, which is this, and oftentimes there's a lot of swelling and edema kind of on either side of the superior sagittal sinus. Okay, I really don't expect you. We're not going to be doing, showing you images on the test of CTs or MRI scans. These are just examples of some things that can go wrong if we have occlusion of the venous system. Okay, finally, let's say a little bit about vascular supply of the spinal cord. All right, so we've already pointed out the anterior spinal artery that, remember, comes off the vertebrals. And the anterior spinal artery continues all the way down the entire length of the spinal cord. Okay, and we have two paired posterior spinal arteries that supply the back of the spinal cord. Okay, but you can imagine that the vertebral arteries, that's not enough pressure to adequately supply your lumbar, your sacral, or your thoracic cord. So the anterior spinal artery needs to have its blood supply boosted all the way down. And so these are the supply is boosted by these radicular arteries. Remember I said a pinched nerve root is a radiculopathy. So when you hear the term radicular, you should be thinking of nerve roots that come out of the spinal cord. And these arteries travel right with these nerve roots. That's why they're called radicular arteries. And so these come off of the vertebral, thyrocervical trunk, intercostal, lumbar arteries. So these radicular arteries are continually given off. And so there is a radicular artery with each nerve root, but it turns out there are only about 7 or 8 that are really significant enough, large enough, to actually supply the spinal cord. Okay, and there's one of these radicular arteries that has a name that is significant here, and I'll tell you in just a minute. All right, so here's the vertebral artery. Okay, and so remember we have an anterior spinal artery here that continues all the way down. Okay, but it keeps getting boosted in the upper portion by the vertebral artery at different segments, and then by intercostal arteries. Okay, so we have these somewhat larger radicular arteries. Okay, so that in looking at the spinal cord, we have one anterior spinal artery that supplies the anterior portion of the spinal cord, just shown here on this side, and then there are paired posterior spinal arteries that supply the back of the spinal cord. Okay, so the one radicular artery you need to know by name, because clinically it's very important. It's called the artery of Adam-Kavitz. Sorry for the difficult name, okay, but artery of Adam-Kavitz. But it boosts the spinal cord supply to the lower thoracic and upper lumbar spinal cord. Okay, why is that important? It's important because there's an area just above it that is somewhat weaker blood supply. We call that a watershed zone. Okay, and so this is an area of the spinal cord that is susceptible to having ischemia, lack of blood flow, from about T1 to T4. And so this can happen, for example, maybe a patient has an episode where their blood pressure is extremely low for a period of time. Or maybe a patient is having surgery on the aorta. And so this, if there's low blood flow, it's the area above the artery of Adam-Kavitz that is susceptible. So this is often where we'll see ischemia of the spinal cord. All right, so here's the spinal cord, and here are nerve roots. So you can see there are radicular arteries here that travel with the nerve roots. But notice here's just a massive, notice this is just a small one. This is just supplying the nerve root itself. But the artery of Adam-Kavitz here, notice how large it is, okay, traveling with the nerve root here. It's a major boost to the anterior spinal artery blood supply. Okay, any questions on vasculature here before we move on to meninges, CSF? Anything else you need labeled? Okay, so let's try a newer PowerPoint format here. Okay, meninges and CSF. So in terms of the meninges, which you will hold in your hand in the lab today and can visualize this, we have the pia mater, which is tightly adherent to the brain and spinal cord. We actually have to peel it off. So it will attach very tightly. We have the arachnoid layer, which extends over all of the depressions and the sulci. Okay, and there's a very important space between the pia and the arachnoid called the subarachnoid space. Clinically, a very important space. Okay, the subarachnoid space coats the entire brain and the spinal cord with CSF, provides this nice protective cushion for the brain and the spinal cord. And there are areas where there are focal enlargements of the subarachnoid space. And these focal enlargements are called cisterns. And there are two cisterns that are important that you know by name, the cisterna magna and the lumbar cistern, okay, which I'll show you here. Okay, so looking at the brain, we have tightly adherent is the pia, and then the arachnoid, and then the dura. Okay, so first a little bit about the subarachnoid space. This contains the cerebral spinal fluid. Okay, and we'll talk about the contents and the function and all of that. And so normally we should just have cerebral spinal fluid there, but there are two things that can go wrong that affect the subarachnoid space. One is if we have an infection that affects the subarachnoid space. That's called a meningitis. Okay, and the blood vessels, which sit kind of right on the pia and into the arachnoid, if we have an aneurysm that ruptures, that blood is going to go into the subarachnoid space. Okay, so that is called a subarachnoid hemorrhage. Right, and so this, again, the subarachnoid space coats the brain and the spinal cord, and at the below the spinal cord, there's a big pocket of CSF in the lumbar cistern. Okay, and so we can put a needle into this lumbar cistern, like at L4, L5, and so remembering that the spinal cord ends at L1, L2, you know that you're safely below that when you do a lumbar puncture, and your needle is just going to go into the lumbar cistern. Okay, obviously, if you don't know, your anatomy and how to find the L4, L5 space, and if you go too high, then there could be some damage that when you put the needle in, you could damage the spinal cord. Okay, so, but we would do a lumbar puncture if we're concerned about a meningitis, because that's how we can evaluate the CSF to look for infection, to find out is it a bacteria, a fungal, or a viral meningitis. And we can also take out cerebral spinal fluid here to look for lots and lots of red blood cells if it's a subarachnoid hemorrhage. Okay, so again, normal contents, cerebral spinal fluid. In pathologic conditions, if we see a lot of blood, it's an aneurysm that ruptured. If we see a lot of inflammatory cells, that would suggest we're dealing with an infection, a meningitis. Okay, so the other space, focal enlargement of the subarachnoid space, is the cisterna magna, which is right here, kind of underneath the cerebellum, dorsal to the medulla. Okay, and in the olden days, there were actually neurologists that would put a needle in here to get cerebral spinal fluid to evaluate for different conditions. We don't do that anymore because it's considered dangerous for obvious reasons, putting a needle right in that location. When I was a neurology resident, actually, there was an older neurologist who would still do these. We call them cisternal taps. He also had a significant tremor, and I just remember watching him, going like this. He was successful. He was very good at it, but anyway, I decided right then I'm never going to do that. Okay, so we've got the pia and the arachnoid. The subarachnoid space is between those two. And then you have the dura, the outer layer. And the dura itself contains two layers. An outer parosteal layer, which is attached to the cranium, contains blood vessels and nerves. And the inner meningeal layer, which is itself attached to the arachnoid. And this is actually important to recognize that we have a lot of nerves in the dura, in the outer parosteal layer. When we talk about migraine headaches later, we will say that's really important in the pathophysiology of migraine, that there's inflammation here and these nerves are activated and involved in the pain of migraine. Okay, now we have separations between the parosteal and the meningeal layer, and this is where the venous sinuses that we were just talking about are located, like the superior sagittal sinus. Okay, really important here. There are two potential spaces that we find either underneath or on top of the dura. So the subdural space is obviously underneath the dura, and then we have a space between the dura and the bone called the epidural space. And this is significant mainly as it relates to head trauma, where we can have blood that either goes into the subdural or into the epidural space. Okay, so here we can see the dura. We have an outer parosteal layer. Remember, that's adherent to the cranium. And we have an inner meningeal layer. And we can see there's a separation here between the outer parosteal layer and the inner meningeal layer. And this is where the venous sinuses are located. Okay, under that we have the arachnoid. And then finally the pia here in purple. And remember, our subarachnoid space is between the pia and the arachnoid. Okay, the subdural and epidural, these are potential spaces that open up when blood is introduced into them. All right, so head trauma can cause bleeding in either of these spaces. So epidural hematomas tend to occur more likely in younger individuals. These are most commonly laceration of the middle meningeal artery. It's arterial blood. Okay, and what's distinctive about epidural hematomas is that the blood wedges into the brain like this. All three of these are epidural hematomas. And epidural hematomas frequently are associated with a skull fracture. Okay, so it's a car accident, something like that, and frequently the patient initially will be awake, maybe complain of a headache, maybe they go home, but as the blood increases here and dissects into the brain, then this can really be catastrophic. Younger individuals don't have much atrophy of the brain, hopefully, and so there's nowhere for that pressure to go. And so it doesn't take much of an epidural hematoma to cause just a catastrophic outcome. I think Liam Neeson's wife had an epidural hematoma after skiing and passed away from that. Okay, so that's an epidural, and there you can see the red arrow there, the skull fracture. Okay, subdural hematomas are much more likely in older individuals. Classically, someone in their 70s or 80s who has a fall. These often evolve more slowly because this is venous hemorrhage. And what happens in a subdural hematoma is you have, we call it shearing of bridging veins. Okay, you shear the venous system, and so blood leaks here into this space, and the appearance is quite different. A subdural hematoma tends to layer out along the entire surface of the brain like this. It doesn't wedge in like an epidural hematoma. And maybe just tell you one thing, just so you know what you're looking at here. There are only two things that can look bright on a CT scan. This is a CT scan here. And the two things that can look bright are calcium and acute blood. Okay. So this is the calcified skull, which is obviously normal. And so this is acute hemorrhage. Okay, the little darker area right here, that means that's blood that's been there for a while. So it gets darker over time. And so here on the subdural hematoma, we know that this happened recently because of all the bright area. The darker area here is, again, it's older blood. Okay, and so you can see this is causing some mass effect here, like this side of the brain doesn't look the same as this side, and so there's some pressure effect there from the subdural hematoma. So again, we have 2 hours on neuroradiology later. We'll go over this in more detail, but I'll just kind of tell you now this would be one of the more high yield images that you'd need to be able to identify for step 1. Right, so the dura covers the whole brain and spinal cord, and we've already talked about these two enfoldings of the dura that really help to stabilize the brain. The falx cerebri, which I had you label on that picture of the venous sinuses, is the reflection of the dura in the interhemispheric fissure. Okay, again, you will hold this in your hands here this afternoon, and we'll talk about it. The cerebellar tentorium is the reflection of the dura in between the occipital lobe above and the cerebellum below. Okay, and the cerebellar tentorium reflects all the way forward. The temporal lobe actually rests also on the cerebellar tentorium, and we'll see that there's an opening at the anterior edge of the cerebellar tentorium for the brainstem to travel through. Okay, so this is another opportunity here to talk about uncal herniation. Right, so here's the cerebellum, and so this dark area right here is the cerebellar tentorium, okay, which reflects, again, between the cerebellum and the occipital lobe, but it extends forward all the way to the temporal lobe. And so what can happen, again, in uncal herniation is the uncus, it gets pushed right over the edge of the cerebellar tentorium. Okay, and when that happens, remember, it pushes on the third nerve and potentially can compress the brainstem as well. So here are some pictures that I'll show you in the lab today. We're looking at the underside of the dura. So this is the falx cerebri right here. So it's in the intrahemispheric fissure. So we've got either side of the brain would be here. This is the superior sagittal sinus right up here. All right, and then this is the cerebellar tentorium back here. So above this, and kind of on the other side, we'd have the occipital lobe. The occipital lobe of the cerebellum would lie on the undersurface of this, and then the temporal lobe. Okay, and again, here's the falx cerebri and the cerebellar tentorium. Okay, and just another look at the falx and the cerebellar tentorium. So this is the edge of the cerebellar tentorium, and this is what I was talking about with uncal herniation. The uncus can just slip over the edge of that. Okay, now let's talk a little bit about the ventricular system. So embryologically, remember we associated each of these cavities with the telencephalon, the diencephalon, the mesencephalon, and so on. And so now looking at the top of the brain, we're just looking at the lateral ventricles. So remember we associate that with the telencephalic structures. And so everything you're looking at here are the lateral ventricles. Okay, now let's just label here the different parts of the lateral ventricles. And they're named by the lobe of the brain that they project into. Okay, so we have what's called a frontal horn of the lateral ventricle right here because it projects into the frontal lobe. We have an occipital horn back here because it projects into the occipital lobe. Okay, and you need to kind of see this as going down into the temporal lobe. So if you can kind of imagine it's going down. This is not obviously a flat structure. The lateral ventricles are another C-shaped structure in the brain. So this is the temporal horn of the lateral ventricle. Okay, everyone got that? Frontal horn, occipital horn, and a temporal horn. Now this large portion right here of the lateral ventricle is called the body. I'm going to just label that. And this area right here, where my circle is called the trigone. And it's called the trigone because from here we go to the occipital horn, to the temporal horn, or up to the body of the lateral ventricle. So this is the trigone. Okay, now looking at it from the side, we'll just label the same thing. So again, everything here is lateral ventricle, so this would be the frontal horn of the lateral ventricle. The body. The trigone. The occipital horn. And now we can really appreciate how this does go down into the temporal lobe. This is the temporal horn of the lateral ventricle. Okay, so that is all telencephalic structures that will associate with that. Now each lateral ventricle empties into the third ventricle. Kind of looks like a bird's head right here. This is all third ventricle. And it gets there through this opening right here. And this opening is called the interventricular foramen of Monro. Okay, so right and left lateral ventricles empty into the third ventricle via this interventricular foramen of Monro. Right? So what is on either side here of this third ventricle? The diencephalic structures. right? So the thalamus is going to be on either side of the third ventricle here. The hypothalamus is going to be on either side of the third ventricle here. Okay, so there are two lateral ventricles, one third ventricle, and one cerebral aqueduct right here. So what are we going to find around the cerebral aqueduct? The midbrain, right? The mesencephalon is associated with the cerebral aqueduct. The cerebral aqueduct continues down into the fourth ventricle, which is right here. And the fourth ventricle, kind of anterior to the fourth ventricle, we'd have the pons and the medulla, and posterior to the fourth ventricle would be the cerebellum. Okay, so the fourth ventricle we kind of see with these rhombencephalic structures. Okay, so I just realized in the break I was looking over the handout, and somehow in your handout the conversion from Word to PDF, some of the labels moved. Okay, so I have this on the PowerPoint slides here. I'll correct the handout here when I have time in a little bit, but because of the shifting arrows, I want to make sure you're looking at this, not the handout. And so let's just go over all of these labeled structures here, and let's follow the production of cerebral spinal fluid from the lateral ventricles, which are here in the dashed line. Okay, there's choroid plexus in all three ventricles, lateral ventricle, third ventricle, fourth ventricle, but most of it is produced in the lateral ventricles. Okay, so we start here. We've got choroid plexus producing cerebral spinal fluid. Okay, so it goes from the lateral ventricles then through this space right here. This is the interventricular foramen of Monroe. And from there it gets into the third ventricle. So this arrow right here would be the interventricular foramen of Monroe. So here's the third ventricle. From the third ventricle it goes to the cerebral aqueduct. From the cerebral aqueduct down to the fourth ventricle. Okay, and from the fourth ventricle the CSF gets out of the ventricular system and into the subarachnoid space. Okay, so we have these outlet channels, little holes here that I'll name them here in just a minute. And from there, again, CSF gets out into the subarachnoid space. So remember, a pocket of subarachnoid space is right here, the cisterna magna. And here we can see the subarachnoid space coating the spinal cord and coating the brain all the way around. Again, subarachnoid space right here. Okay? So CSF circulates around the brain as the subarachnoid space, and then it gets out actually into the superior sagittal sinus. So this right here is the superior sagittal sinus. And so CSF gets from the subarachnoid space into the superior sagittal sinus through these openings here, which are called arachnoid granulations. Okay, and it turns out that the pressure in the subarachnoid space is just slightly higher than the pressure in the superior sagittal sinus, and so therefore the flow of CSF is just continuous and easily empties into the subarachnoid space. And so we can follow this down. Here's the the superior sagittal sinus, okay, which joins with the straight sinus into the sigmoid sinus and out the internal jugular vein. So the venous system here, these venous sinuses, not only convey venous blood out to the internal jugular vein, but also all of the cerebral spinal fluid as well. Okay, so again, on a blank drawing, you know, after you spend some time, see if you can label all of these structures here on that one. So from the fourth ventricle, I mentioned there are these outlet channels here in the fourth ventricle. Two of them are lateral, the lateral foramen of Luschka. So here we can see on this side and over here. So again, the CSF gets out into the subarachnoid space, coats the brain and spinal cord. And then there is a midline foramen of Magendie, which is kind of a midline outlet channel from the fourth ventricle. Okay, and as I mentioned here, this is subarachnoid space. Okay, again, between the pia and the arachnoid. Okay, and so through these arachnoid granulations, the cerebral spinal fluid is able to empty into the superior sagittal sinus. All right, so what's in the CSF? We said it's produced by choroid plexus. Important that you know, I mean, if you're going to do a lumbar puncture and analyze CSF, you need to know what's there and what shouldn't be there. And what shouldn't be there are any red blood cells or white blood cells. It's also important to know that glucose in the CSF is 2/3 that of blood. And routinely, when we do a lumbar puncture, we always also at the same time check the blood glucose. Okay, it's important to compare the two. So CSF glucose is 2/3 that of blood, and there's relatively little protein in the CSF, 0.5% that of blood. So the function of CSF, we've said, is that just the brain literally floats very nicely here in this subarachnoid space, also cushioned by the ventricular system. So there's a protection and a buoyancy. It also filters out a lot of toxic substances. And just recently there's been this type of lymphatic system, which are perivascular pathways where CSF actually circulates through brain tissue. And this seems to be significant mainly because it filters out amyloid beta. Okay, when we talk about Alzheimer's disease, this is bad. You don't want to have amyloid beta accumulating. That's related to Alzheimer's disease. And so CSF is actually able to filter out some of these toxic substances. Okay, let's finish just talking a little bit about hydrocephalus. Okay. Hydrocephalus, well, just first normally to appreciate, the choroid plexus makes about 500 cc's of CSF per day. Okay, but at any given moment, the total CSF in both the ventricular and the subarachnoid space is only 150. Okay, what that means is this needs to circulate very efficiently. And if it doesn't, if there's a blockage, then quickly we can develop hydrocephalus. And there are 3 types of hydrocephalus. The first 2 are true hydrocephalus, non-communicating and communicating. This is kind of a pseudo, sort of looks like hydrocephalus, but it really isn't. Hydrocephalus ex vacuo. All right, a non-communicating hydrocephalus is where there isn't communication between the ventricular system. We don't have communication between the lateral ventricles or the third ventricle or the fourth ventricle. So just as an example, let's say we have something occluding the cerebral aqueduct right here. Maybe it's a tumor, or we just have a narrowing of the cerebral aqueduct. Well, what's going to happen? You've got CSF being produced by the lateral ventricle and the third ventricle, and it has nowhere to go. So the lateral ventricles and the third ventricles will dilate. Okay, that's hydrocephalus. And since it's a non-communicating, we don't have communication between the third and the fourth ventricle. This is an example of a non-communicating hydrocephalus. And so here's a patient that has a tumor lesion around the cerebral aqueduct, and you can see the lateral ventricles just dilate. Okay? And so very, very important. We do lumbar punctures for a lot of different things, but if a patient has a non-communicating hydrocephalus, it is dangerous to do a lumbar puncture. And the reason is, okay, you put a needle in the lumbar cistern, you drain out a bunch of fluid to send it to the lab. And what happens is you are creating a pressure gradient between a very high pressure here and now a very low pressure down in the lumbar cistern. And so the entire brain can herniate in that situation. And so generally, not 100% of the time, but most of the time before we do a lumbar puncture, we like to do a brain scan just to make sure that we're not dealing with something like this. Okay, so do not do a lumbar puncture in a case of a non-communicating hydrocephalus. A communicating hydrocephalus, an example of that would be, let's say we have scarring of all of the arachnoid villi up here. And I'll tell you later conditions that can do that. All right? Now the ventricular system communicates, right? That's not a problem. But there's just a lack of getting all of it out into the superior sagittal sinus. All right? So that would be a communicating hydrocephalus. And so the ventricles would look big. Just looking at this, I couldn't tell you whether it's a communicating or a non-communicating hydrocephalus. But in a non-communicating, in a communicating hydrocephalus, you actually can safely do a lumbar puncture because you don't end up creating a dramatic pressure gradient. Okay, so let me just finish here on this slide. This is normal, let's just say, normal appearing ventricles. This would be a hydrocephalus. You've got large ventricles, and you couldn't tell from this, is it communicating or non-communicating? I don't know. We'd need, the rest of the brain to look. But this is the third type of hydrocephalus, and it's called hydrocephalus ex vacuo. And what has happened here, it's really not a hydrocephalus. Notice all of the dark area around here. What we're looking at here is atrophy of the brain. Okay, this is what happens in Alzheimer's disease and many other neurodegenerative conditions where the brain shrinks, okay, including the tissue around the lateral ventricles. So the ventricles look bigger, but they're not under pressure. Okay, this is not really a true hydrocephalus. They just have the appearance that they're larger. And so at autopsy, you can just see a lot of atrophy of the brain everywhere, both centrally and peripherally. That's called hydrocephalus ex vacuo. All right, so we'll see you in the lab this afternoon. Make sure you have your lab coat, and we have the whole class here meeting at 3 o'clock.